Hey guys, so today we are going to be doing the last sensory organ. We've already gone over briefly the nose, mouth, and skin, and then in the last couple lessons we looked at the eye, and today we're going to be looking at the ear and how that all functions. So in this lesson on the ear, we're going to be going over the two different functions of the ear. We're going to be describing the three sections of the ear, as well as all of the structures within each section and the function of each of those structures. We're going to talk about the pathway that a sound wave will travel through from the outside of the ear to the sensory receptors. We're also going to talk about how those sensory receptors send an impulse to the brain and how it's interpreted by the brain. And then we're also going to talk about how the ear helps with head positioning and with balance, because the ear is not only responsible for hearing. So the two main functions of the ear are hearing and equilibrium. So you have mechanoreceptors, which are called hair cells, in the inner ear, which converts energy from sound waves, which is kinetic energy since sound waves move, into the electrochemical energy that is used to generate that action potential. The mechanoreceptors, which as I said are called hair cells, and essentially they move or are bent when the sound wave hits them, essentially. And the bending of the hair cell is what stimulates the hair cell to start depolarizing and then send that action potential. Mechanoreceptors called proprioceptors are also found in the ear. They also convert kinetic energy into electrochemical energy. Okay, there are three different sections of the ear: the outer ear, the middle ear, and the inner ear. And there's a bunch of different structures in each of these. So the outer ear is made up of the pinna, the auditory canal, as well as earwax. The pinna is just the external flap of ear, and its whole job is to kind of collect sound and funnel it into the auditory canal. The auditory canal is then the hole in your ear, which directs any incoming sound waves to your eardrum. And then you also have earwax in there, which is secreted by specialized sweat glands, and their job is to trap any foreign particles and stop it from entering into the ear. So it's essentially trying to prevent ear infections and stuff like that. Then we get to the middle ear, which starts with the eardrum. The eardrum is also called the tympanic membrane, and essentially what happens is the sound wave travels down the auditory canal and then smacks into the eardrum, or the tympanic membrane. And when that sound wave hits the eardrum, it's going to kind of vibrate the eardrum. And then behind the tympanic membrane, we have an air-filled chamber that has three tiny little bones in it called ossicles. And those ossicles are attached, one of them is attached to the eardrum, and then they're all attached like in a chain, one after the other. We have the malleus, incus, and stapes, also called the hammer, anvil, and stirrup. You don't have to know their individual names, you just need to know that they're the ossicles. And then the last of those three ossicles attaches to another membrane called the oval window, which connects the middle and inner ear. So the sound wave is going to hit the eardrum, the tympanic membrane, cause the vibration to travel through the tympanic membrane, through those three little ossicles, and then the last of the ossicles is going to vein into the oval window membrane and cause that to kind of push inwards. The middle ear also has something called the eustachian tube, which moves air between the middle ear and the mouth. And the whole point of that is to keep the air pressure in the middle ear kind of similar to atmospheric air pressure. That's why, like, if your ears are ever like popping or something, you might like yawn or open your mouth really wide to try and get your ears to pop. That's because the eustachian tube that regulates that kind of air pressure in your ear is attached to the back of the mouth. So yeah, sound vibrations hit the eardrum. The waves are going to move through the three ossicles, and then the tympanic membrane, the eardrum, is much larger than the oval window membrane. And so as the sound wave goes from the larger membrane through the ossicles to the smaller membrane, that sound is concentrated down. And then because the sound is concentrated, that means that it's also amplified. Then we get to the inner ear. The inner ear has 3 distinct structures: the vestibule, the semicircular canals, and the cochlea. The vestibule is 2 little sacs right next to the oval window. Those 2 little sacs are called the utricle and saccule. They help determine head positioning. So like if your head is like tilted, for example. These semicircular canals are 3 semicircular tubes, all arranged at like slightly different angles. And as fluid moves through those canals, it helps for you to identify if your body is moving or if your head is moving. And then neither of those are actually connected at all to hearing. They have nothing to do with hearing. They have to do with equilibrium, with balance, with positioning. The only part of the inner ear that has to do with hearing is the 3rd distinct structure, the cochlea, which is like a little spiral snail shell shape, and it's going to contain a bunch of hair cells. Those are the hair cells that respond to the sound wave. They're stimulated when that wave comes and causes them to bend. That is what stimulates them to depolarize and send out their nerve impulses. So this is just kind of an image of these different parts. So out here we have the pinna and the auditory canal. Those are the 2 parts of the outer ear. Then in the middle ear, we have the tympanic membrane, aka eardrum. These 3 little bones in here are the ossicles. And then this little circle right here at the end would be the oval window. And then this eustachian tube is regulating the air pressure of that middle ear. And then we get to the inner ear, which has the cochlea, which is this kind of spiral here. That's the hearing portion of the inner ear. We also have these semicircular canals. You can only see 2 from this angle, but there's actually 3. And then these 2 kind of like bulgy parts right here, that's the vestibule. So the saccule and utricle make up the vestibule. Okay, inside the cochlea, we have something called the organ of Corti, which is the middle chamber of the cochlea. Within this organ of Corti, you have the basilar membrane across the bottom. Attached to the basilar membrane and kind of sticking up off of it are the little hair cells. On top of the hair cells are stereocilia, which are just little tiny projections. And the tops of those stereocilia are attached to another membrane called the tectorial membrane. What's going to happen is the sound wave is going to come into the cochlea. It's going to hit the bottom of the organ of Corti, so it's going to hit that basilar membrane. That's going to push the basilar membrane upwards, which causes the hair cells to, of course, move upwards. And it causes the little stereocilia on the top of the hair cells to bend against the tectorial membrane. So it's essentially like, if you picture, I need something that's kind of bendy. Okay, so pretend that this pink sheet here is your hair cell with like stereocilia on top. And so underneath here, I have the basilar membrane, which is my lower hand. And on the top here, I have the tectorial membrane. And so when the sound wave comes up and hits the basilar membrane, it's going to, whoopsies, it's going to bend the hair cell against the tectorial membrane. So the tectorial membrane is not moving, the basilar membrane comes up and it bends the hair cell and there's stereocilia on the top of the hair cell against that tectorial membrane. It is that bending of the hair cells that is going to, or the stereocilia on the hair cells, that is going to stimulate the hair cells to depolarize and send out their impulse. So this is what it looks like. This is a cross section of one of the like tubes that are like spiraled around in the organ of Corti. And as you can see in the middle chamber there, we have the basilar membrane along the bottom, the tectorial membrane along the top, and the hair cells in the middle. If we just kind of zoom in over here, again, this is the basilar membrane on the bottom, the tectorial membrane on the top. These are the hair cells, and the little black things are the stereocilia. So what happens is the sound wave is going to come in, it's going to push upwards against this basilar membrane, which pushes these hair cells upwards. Their stereocilia gets bent against the tectorial membrane, and the bending of those stereocilia stimulates the hair cells to depolarize and send out their impulse. Okay, so this, I wouldn't actually like memorize it like this. I would memorize it with like a diagram of the ear. But this is just kind of showing the pathway of sound. So again, the pinna is going to funnel it into the auditory canal. The auditory canal directs the sound to the eardrum. The sound hits the eardrum vibrates. That vibrates through the ossicles. The last ossicle hits the oval window. The oval window then pushes the sound wave inwards. It's going to cause the fluid in the cochlea to start swirling through the cochlea. That is then going to push against the basilar membrane in the organ of Corti. That basilar membrane getting pushed upwards is going to cause the hair cells to get pushed upwards. Their stereocilia bend against the tectorial membrane, which stimulates the hair cells to depolarize. Your action potential is generated, your impulse is sent. The impulse is going to get sent along the auditory nerves, which then carry that impulse to the temporal lobe of the brain. Okay, so this is just another diagram of this. So again, the pinna is going to funnel sound into the auditory canal, which funnels sound towards the tympanic membrane, aka eardrum. That is going to push inwards and vibrate the three ossicles, which then hits the oval window right here. That oval window pushes inwards, which causes the fluid in the inner ear to get pushed through the cochlea, and then inside that cochlea, you're going to have the basilar membrane pushed up into the hair cells, bent against the tectorial membrane, etc. You can also see the eustachian tube here, which again is regulating the air pressure in this inner ear here. You can also, from this angle, see all three of these semicircular canals. And then you would also have in this area here would be the vestibule. You can also see your auditory nerves, which are coming out the back here. And does anyone else think that this looks like a chameleon? Is that just me? With like its tongue sticking out? Okay, frequency refers to how close together the sound waves are. Higher frequency means the sound waves are closer together. Lower frequency means the sound waves are further apart. If a sound has higher frequency sound waves, that means we will interpret it as a higher pitched sound. High frequency sounds vibrate the basilar membrane closer to the opening of the cochlea, whereas low frequency sounds will hit the basilar membrane closer to like the inside, like tight spiral part of the cochlea. And so depending on where the impulse is sent from, that's going to be interpreted by the temporal lobe as either high-pitched or low-pitched. Amplitude is then the height of the sound wave, so like a higher amplitude or a lower amplitude. If you have a higher amplitude sound wave, it's going to be interpreted as a louder sound, which then is just going to cause the hair cells to bend more, which causes more of them to send off impulses. So we detect this using summation, which if you remember is the more impulses that are sent, the stronger the stimulus we interpret. The muscles that hold the muscles that hold the ossicles together will contract anytime sound is really intense in order to try and stop the ossicles from vibrating around too much. However, if you have a lot of really loud sounds pounding into your ears all the time, those high pressure waves can actually come in and damage the inner ear and can actually cause permanent damage to the stereocilia. There's little tiny little things on top of the hair cells that are getting bent against the tectorial membrane all the time. And that can eventually result in hearing loss. That's why you're supposed to, you know, not crank super loud music in your headphones, or like wear ear protection when you're mowing lawns. There are two main types of hearing loss. The first is conductive hearing loss, where the sound waves are just not reaching the inner ear properly. And this is where hearing aids can be used to amplify sounds and transmit them to the eardrum. But then there's also sensorineural hearing loss, which is where like the hair cells, the stereocilia, or the auditory nerves are damaged. And at that place, in that case, you actually need cochlear implants in order to kind of bypass that damaged area and convert sound waves into electrical impulses. In that case, the cochlear implant doesn't actually replicate the exact sound, but it does provide sounds that allow the person to actually interpret their environment. And over time, the person can learn to interpret those impulses to actually understand speech. Because as we talked about with like, you know, the glasses that flip your vision upside down, your brain is really adaptable, and the younger person is the more adaptable or elastic, how it's referred to, their brain is. Okay, so that's it for the hearing portion of the ear, but as we talked about, the second big function of the ear is with equilibrium, which is essentially like your balance and stuff like that. And your balance is made up of two different types of equilibrium. The first is called static or gravitational equilibrium, which is essentially determining the position of your head relative to gravity. So is your head tilted, or are you like lying down or stuff like that? And then there's dynamic equilibrium, which involves determining which way the head is moving in 3D space. So that could be if like you're moving your head on your body, or if like your whole body is moving and therefore your head is also moving because it's attached to your body, like if you're spinning on a chair or something. The static equilibrium is determined in the vestibule. So the utricle and saccule, those are fluid-filled sacs that monitor how your head is tilted or positioned relative to gravity. Inside of these sacs, you have tiny little hair cells with cilia that stick up into the jelly that is filling this sac. And then floating on top of the jelly are little granules of calcium carbonate called otoliths. And what happens is when you tilt your head, for example, those otoliths are going to shift because of gravity. And when the otoliths shift or fall, they're going to kind of drag the jelly with them. And as the jelly moves, the hair cells that are sticking up into the jelly get bent by the jelly that's kind of like flowing past. And once again, it is the bending of those hair cells that stimulates them to depolarize and send out an impulse. In this case, the impulse gets sent to the cerebellum. So this is kind of what it looks like. So this would be like inside of the utricle or saccule, one of those sacs. So we have the hair cells here with the little stereocilia sticking up. This kind of yellowish area is the jelly, and then you have the otoliths, those little like granules on top. And then if the person tilts their head, the otoliths are going to fall down due to gravity, so they're going to kind of slide downwards. That pulls the jelly with it, and that's going to bend those little hair cells that are stuck up into the jelly, and that stimulates them to depolarize. And then dynamic equilibrium, which is the rotational equilibrium, is determined by your semicircular canals. So you have 3 semicircular canals. Each one has an ampule, or like a little pocket or bulge at the base of it. And inside that ampule are hair cells. The hair cells have little cilia on them again, and they are contained inside a cupula, which is like a little helmet that goes over top of the hair cells. And what happens is when your head rotates either on your body or with your body, that is going to cause fluid to like roll through these semicircular canals. And as the fluid goes through the semicircular canals and then comes back around to the base, it's going to flow through the ampule. And as the fluid goes past, it's going to push the little like cupula with the hair cells in it. And again, the bending of those hair cells is going to be what stimulates them to send an impulse. Just like with the static equilibrium, it is interpreted by the cerebellum. This is also what causes motion sickness. For those of you that get motion sickness, it is essentially your semicircular canals sending out all of this information that, you know, your body is moving, your head is moving a lot, especially if you're not interpreting that movement very well visually and you're just feeling the movement. That can cause a lot more motion sickness. But yeah, motion sickness is essentially just this fluid really violently moving through your semicircular canals. I picture it like a roller coaster, like the semicircular canals are like a roller coaster for the fluid. And yeah, that's why it's like motion based and gets you sick, if you're like me. So this is what it looks like. So this whole thing up here is the entire inner ear, so the cochlea, which is the hearing part. Then these kind of like this bulgy area at the base of both of these, which the utricle and saccule, those are the vestibule. And then we have the 3 semicircular canals. And as you can see right here and here and here, there's this little like bulgy area at the bottom of each semicircular canal. That is the ampule. And inside the ampule, you have the hair cell here at the bottom. It has its little cilia sticking up inside this big white helmet, which is the cupula. And so what'll happen is the fluid is going to rush through this semicircular canal, and then as it comes down through the ampule, the fluid rushing through here is going to push on that cupula, and the bending of that cupula and the stereocilia inside of it is going to stimulate the hair cell. Okay, let's switch over to my document cam for one last picture here. Okay, so this is just one last diagram of the ear here. So again, we have the outer ear, which is made up of the pinna and the auditory canal. Then the middle ear, which is made up of the tympanic membrane, the 3 little ossicles, and then the oval window, as well as the eustachian tube. And then the inner ear, which is made up of the cochlea, the semicircular canals, and the vestibule, which contains the saccule and just to kind of summarize the different areas of the ear here, we have the hearing portion of the ear, which is made up of the outer, middle, and part of the inner ear, and then the equilibrium portion. So for the hearing portion, again, the outer ear is the pinna and auditory canal, the middle ear is the tympanic membrane, the ossicles, the oval window, and the eustachian tube, and the inner ear is the cochlea, which contains the organ of Corti, and inside the organ of Corti, we would find the basilar membrane, the hair cells, and the tectorial membrane. By the way, all of the equilibrium stuff is also in the inner ear, as well as the cochlea. Of all of these different hearing structures, the only one that sound waves don't actually travel through is the eustachian tube. All of the rest of these are kind of like in order of how the sound waves would go through them. And then with the equilibrium portion, we have either the gravitational, aka static equilibrium, or the dynamic, aka rotational equilibrium. The gravitational equilibrium is controlled by the vestibule. The vestibule is made up of the saccule and utricle. Inside of the saccule and utricle, we find the hair cells, the jelly, and the otoliths, and this all helps determine head position. And then in the dynamic equilibrium, that is controlled by the semicircular canals. There are 3 of them. Each semicircular canal has an ampule at the bottom. Inside the ampule is the hair cells, the cupula, and the fluid, and this determines head movement. Okay, so let me know if you have any other questions about this stuff.